Hello everyone. Today we're going to be looking at the phenomenon known as neural communication. This is a basic process of this video is going to help explain the process of how a nerve cell is receiving a stimulus and then sends that signal on to any other adjacent cells. These adjacent cells can be anything from other nerve cells to muscle cells to wherever the nerves happen to lie to send that stimulus on so that the target cells can then do what they need to do. So first, what we're going to look at is we're going to look at the anatomy of a neuron. It's important to know the structure of this of the cell that we're talking about so we can see how it functions in a very basic manner. We also need to look and see how this is going to happen in a much more detailed description. So we're going to look at a phenomenon known as action potential. We're going to look at how this action potential happens on the most basic level. We're going to go into a little bit of in-depth analysis on how this action potential occurs. And one of the things I also like to do is also look to see this is how the function of the cell should work. But one of the things I always am fascinated by is, let's see what happens if this cell doesn't function under ideal conditions, and what are the effects of the cell not functioning in those ideal situations and good circumstances, and what are the effects afterwards, and what is it that is causing those effects to happen, and what can that lead us to? Are there other larger implications? So first we're going to look at the anatomy of a neuron. Here we go. So looking at the anatomy of a neuron, this is one example of a neuron that we see right before us. We have the dendrites over to the left, the nucleus of the cell, which is right there as we can see in the middle. We're going to then look at the business end here with the axon, and in the axon we have both the Schwann cells, which are providing sort of protection and insulation, and in between those Schwann cells, the myelin, we have the nodes of Ranvier. At the end of this, of course, is the axon terminal, which is going to send the message to those neighboring and adjacent cells. Now, with this particular example that we have the myelin sheath and the nodes of Ranvier, and what we get here is a very fascinating occurrence called saltation. This allows the saltation process allows the message being sent from the dendrites through the axon to happen at a much faster rate. And this saltation occurs in a fascinating process, and not only just neuron cell saltation as a process of bouncing along. See this in geology along the bank of a river, where sediment is being transported, and it's doing so as it bounces along. In the case of biology, what we're looking at, especially in a neuron, again, we get this bouncing motion going over the parts of the cell, and what we're seeing is that is allowing an increase of the signal to the time is shortened so that the message can be sent that much faster. Think of the most basic example I can think of is the message is parkouring over the myelin and traveling through the axon to get to the destination down the axon terminal and to all other adjacent cells that it needs to get to produce the desired effect that is being sent by the stimulus. So let's look at resting potential here for just a moment. The neuron is going to stay at resting potential whenever it is not receiving a stimulus. Inside of the cell, the neuron has a negative charge. And outside of the cell has a positive charge. And they've been able to measure this electrically, and they've found that the charge inside the cell is at negative 70 millivolts. And of course, this will change once we receive the stimulus, and we're going to go into that action potential. So let's look at a very brief overview of that action potential and go on from there. So the action potential, very simple explanation here. We receive a stimulus, dendrites are the ones receiving that signal, and they're going to pass that on from the cell body down to the axon, and that message is going to continue on down the axon to the axon terminal, and then it's going to go to all of the adjacent cells. These cells, again, they can be muscle cells, which will then cause you to contract your muscles. These could be more nerve cells that are sending a signal a little bit further along, and they can be other cells within the body, again, providing that desired response. Important to note, and again, fascinating, that we always talk about synapses and so forth firing, and they do so electrochemically. And we have evidence of that as we have just shown. So again, starting and remaining at this resting potential until we receive a stimulus. We have a wonderful graph here to look at. Now, once this change has occurred, what we're going to get here is the cells are going to receive a stimulus. We have these voltage-gated channels, which you can see here. This one is for sodium, and this is for potassium. These are very simplified diagrams. So we open up the voltage-gated channel for the sodium, and it allows the sodium to rush in, and this change, of course, is going to change the charge within the cell from negative to positive. And you can see that change is going to go from negative 70 volts, and the graph that we have here up to 30 millivolts, and that is going to continue to propagate. So the sodium rushes in, and as that sodium rushes in, it's also going to cause voltage-gated potassium channels to open, and the potassium is then going to go outside of the cell. Once those cells, the potassium ions are expelled outside of the cell, the sodium travels to the right, and the sodium is going to continue on. It's going to activate more sodium-gated channels, allowing more sodium to rush in, and again pushing that potassium out to continue to propagate that signal. And we're going to continue to do this until we get to the axon terminal, and eventually we're going to get to the end of the message. Now, as we're doing this, action potential is very quick and very brief. So a quick explanation. Again, we open up the sodium-gated channel. Sodium rushes in, potassium rushes out. As this happens, the sodium again is moving through. Once it reaches this point, this is a sodium-potassium pump, and what that sodium-potassium pump is going to do, it's going to help repolarize the area that has just sent this signal. So it's going to have the sodium go out, and it's going to have the potassium go back in. It's going to restore these levels back to where they were before. And this is going to then repolarize the area, and then we're going to go back to resting potential. So let's look at that again, just from one more perspective. We have a simple example here of a neuron. In this case, it has no myelin sheath, but quick explanation here. We have this area becoming depolarized, the process that we just mentioned. As that message goes to the right, we then have that area immediately repolarizing so that it would be ready to receive another stimulus once it has gone back to resting potential. And this process repeats itself all the way down until we get to the axon terminal and send the message to all those adjacent cells that have been mentioned previously. Now, again, this is how this is supposed to work. This is optimal conditions. Some recent studies have been looking at ways that are affecting how this signal is transmitted, and of course, one of the things that they have looked at is the good old substance alcohol. Alcohol binds to nerve cells, and once it does so, it allows the nerve cells to grab a lot more chlorine ions, and the chlorine ions, of course, would then be negative, and those negative ions are going to go inside that cell, which is going to make the cell more negative. which means that when we're receiving a stimulus, we're going to need more sodium to allow that message to propagate, which is going to slow down the entire process. There are videos of this showing how quickly the cell is receiving a signal, but how slow it is to have that signal pass on, because we've put in a lot more negative ions and are therefore slowing down the process, because in order to depolarize this area, we're going to have a lot more positive coming in. Once that happens, it's going to be the same process and has a cascading effect all the way down from one axon terminal to the next. This is why we're so slow, and our reflexes are way down when we indulge in alcohol. The study that I was looking at specifically focused on adolescents. They were looking at adolescent rats, and not only did they discover that the message was slowed down, but they also discovered that in a developing brain, we also see that the alcohol, because it's a drug and because it releases other things like dopamine, that it is also possible for that brain to become addicted, especially at younger ages. They did test older rats as well with some of the same conditions, and they found that the older rats did not develop a dependency on alcohol, whereas the younger or adolescent rats did the same. And since we share that similar biology as far as nerve cells and just basic body structure, this can very easily extend to humans as well. Always fascinating to see this on a microscopic scale, and then see how it affects the entire body. A lot of times we neglect to we look at the micro and neglect the macro, or sometimes we look at the macro and don't think about the micro. It's always important to remember that these things work in tandem and have some serious effects throughout. Anyway, here are the references to the information that we've just given. You guys can examine these references at your leisure. I hope you've enjoyed the video and that we've learned a little something together.